Good morning, everybody, and welcome to this Cryo EM virtual roundtable organised by Quantifoil and EM Japan jointly. We're very pleased to have you join us, and very pleased indeed to have three highly experienced Cryo EM experts joining us this morning to discuss the most exciting aspects of Cryo EM currently. So I will just share my screen and show you our agenda for the morning. For the afternoon for you, apologies. Just 1 second. There we go. It's just taking a little while to share the screen. Could I just ask one of the panelists if they can hear me?
Yeah.
Yeah.
Okay, my controls have paused. That's the problem. So I will talk you through it and then move you across to our first presenter. So this morning we have three very experienced Cryo EM experts. We have Professor Koji Yonokura from the Spring 8 Center at Riken, Professor Radostine Danav from the University of Tokyo, and joining us from the University of York, Dr. Jamie Blazer. The plan this morning is to speak first with Professor Koji Yonokura to speak. Professor Koji Yonokura has worked for many years in Cryo EM in the biomechanical structures group in Riken. He has worked also extensively on methodology and technology developments to aid in the ease and efficiency of Cryo EM for both for SPA, but also looking at microelectron diffraction for smaller molecules, which is becoming increasingly popular. So I'd like to hand over to Professor Koji Yonokura to give us a more thorough introduction into his work and what he's been doing. And Priya, can I ask you to change the presenter because my controls are currently non-responsive?
You'll be able to share your screen.
Okay.
Now.
Yeah.
Okay. Can you see my display, my screen? Can you see? No.
Yeah, I can see.
Okay. Good.
Yeah,
Good.
I have a problem with my controls.
So I think it's ready. Okay. Okay. Thank you. Okay. Oops. Just a moment, please. Okay. Oops. Okay. So I'm Koji Yonokura at Riken Spring Center and Tohoku University. Today I'm going to talk about two-way use of Cryo EM for higher resolution single particle analysis and high precision electron 3D crystallography. So the subtitle of my talk also is revealing hydrogen and beyond. Okay, let me get started. So we have been using quantified Holy carbon film for more than 22 years. Thanks. Okay, so it is the year that your company was launched. So I think it is a really good items, but delivery is always slow. So please improve this point. Okay, let me introduce our Cryo EM activity. So we have designed a new Cryo EM system for the two-way use. So then we have installed a JOL cryo 300 electron microscope as the first machine of this model. The microscope is operated at an accelerating voltage of 300 kilovolts and equipped with a cold field emission gun, an incurrent energy filter, and we have 3 high-spec cameras. It also supports automated liquid nitrogen filling and control by serial EM. So operation of this system started in July 2018. Despite initial flaws and bugs, as can be expected with any new machines, the system is now much improved and has successfully yielded high-quality data in both applications. So one highlight in this Cryo EM is cold field emission gun. These are theoretical CTF curves. Red represent curves for cold field emission gun. Blue, thermal field emission or Schottky emission, which is used in the most high-end Cryo EM nowadays. The shape of this curve is very important, as in free space, structural factors are essentially multiplied by CTF. And in real space, FFT of CTF becomes a point spread function of EM. So this is a zoom up view of the higher resolution region. So as you can see, CTF curve of the CFEG is less attenuated. Thus, cold field emission gun can improve signal in super high resolution beyond 1 angstrom resolution. So these are FFT or metal thin film images taken with a normal Schottky gun and with CFEG. As you can see, CFG gave remarkable better signal over the Schottky emission. The CTF ring patterns are very clear, even beyond the 2 angstrom resolution, and still visible at about 1.4 angstrom resolution. This is another example again, metal film. It resolves CTF rings patterns further to 1.13 angstrom. Important thing here is that data were collected in the same condition as single particle analysis of protein samples. Okay, using this system, we have obtained a 1.5 angstrom Cryo EM structure of a protein complex, apoferritin, in February 2019. It was the highest resolution single particle reconstruction at that time. Then we published several papers as a first study showing the power of CFEG in the Cryo EM. Okay, now we determine the structure to sub 1.2 angstrom resolution. This is a tyrosine residue at a higher display level. You can clearly see individual atoms here, and also you can now see many hydrogen atoms here. Furthermore, but I'm sorry, I can't tell more today. I'm so sorry. But this is a comparison of scattering factor of hydrogen atom for X-ray and electrons. Here, amplitude scaling is adjusted at the origin of the X-ray and electron scattering amplitude of the carbon atom. And so here, relative amplitude of are displayed for comparison. The hydrogen atom shows higher scattering factor for electrons than X-ray. Thus, electron scattering can resolve hydrogen atoms well, thanks to partly this. So of course, the absolute scaling of electron scattering is 4 to 5 orders of magnitude more stronger than that of X-rays. So I picked up this table from a very famous Richard Henderson paper. And we have solved many interesting structures so far by both single particle analysis and electron 3D crystallography. Some of them have been already published And let me introduce these results. These people in my group are doing sympathetic cryo EM. Tasuku is a player for this. And also, Keisuke is a good at model building and analysis of the model. And the first example is a membrane protein complex, photosystem 1. This is a 2.58 angstrom resolution structure of PS1 from a cyanobacteria. It forms a homotrimer. This cyanobacterium uses far-red light or low-energy light for oxygenic photosynthesis. And so how low energy light can be used for photosynthesis has been a long-standing question. And the PS1 has many pigments or cofactors. Thanks to high-quality Cryo EM map, we can see many of those cofactors. Interestingly, all other organisms use chlorophylls, but interestingly, we found that this cyanobacterium has a pheophene where major magnesium at the center is missing, as shown. We think this is a key how this cyanobacterium can use low energy light for photosynthesis. And we recently solved another PS1 structure at about 2.0 angstrom resolution, as shown here. It clearly resolves individual atoms, including the magnesium as the chlorophyll center. The quality of the map is beyond the X-ray crystal structure of these molecules. Let me move to another example. It is a nucleic acid binding protein. You can see many crowded molecules inside amorphous size. These high contrast ones are contamination, not protein. Protein molecules are surrounding particles. And then we calculated the 2D class averages and these 3D classification. Hamaguchi-san eventually obtained two 3D reconstruction representing different conformations, as shown here. And Hamaguchi-san and Kawakami-san then found large domain motion upon nucleic acid binding, as shown here. So these are atomic models based on the Cryo EM maps. Okay, let me move to electron 3D crystallography. Kaori and Kiyofumi are doing this in my group. And we also usually use quantifiable grids for electron 3D crystallography. We often obtain very small crystals. These are membrane protein crystals. These crystals are too small for X-ray diffraction, even with a high-intensity synchrotron radiation beam. In contrast, electrons often yield beautiful diffraction spots beyond 2 angstrom resolution. As I said, this is because electrons are scattered by light atom 4 to 5 orders of magnitude more strongly than X-ray. We can now solve high-resolution structures from these crystals of various samples. But this technique is recognized as being more important in synthetic chemistry, material sciences, and related areas because single particle analysis can be used for larger size protein and protein complexes. More importantly, electron diffraction is directly related to Coulomb potential, where X-rays are scattered by electron clouds and atoms. So with electron diffraction, we could see Coulomb potential or charged state of molecules. Hopefully, this would lead to better understanding of working mechanisms, structure formation, stability, and so on. So our system, Cryo 300, has an in-column type energy filter, which can remarkably reduce background noise as shown here. Please compare these electron diffraction patterns. Also, signal-to-noise ratios or I/sigma values of individual diffraction spots were significantly improved even in 3 angstrom resolution range by energy filtration. Then we solve 3D structure of protein crystal from rotational electron diffraction patterns, as shown here. An important thing is that only elastically scattered electrons represent accurate Coulomb potential. And this is a Coulomb potential map of the heme binding site of catalase, thus solved by molecular replacement from these rotational electron diffraction patterns. Error factors or R factor values are comparable to those by X-ray crystallography. So this result shows the high performance of our Cryo EM system. There have been many proof of principle studies so far, but little for application to unknown 3D structure of protein complexity of complex molecules. So our group focuses on difficult target crystals as well as development of technologies. So these are examples of structures we have determined so far. None of them had been solved before. Let me introduce these. And these are polypeptide structures. We solve the two structures, wild type and point mutation. The mutation causes a serious neurological disease. And in the mutation, we found many hydrogen bonds are formed, which would promote toxic aggregation. And this is a new double helix structure of nanographene molecules. I think this is one of the most complex organic molecular structure solved by electron diffraction. And this beautiful structure would become a base for designing functional nanographene assemblies. And this is a high resolution structure of an organic semiconductor material. We can clearly distinguish carbon and sulfur atoms in this high-resolution 3D map. And these are drug candidates. We solve this structure from less than 0.1 milligram sample. In the left figure, we found the structure in the crystal is different from a predicted one by calculation. And in the right figure, we found that the crystal is composed of racemic mixture of this molecule. This was unexpected. And this is another demonstration for pharmaceutical application. It is ivermectin or ivermectin in Japanese. So this molecule is is a good candidate for fighting COVID-19. And for high precision analysis, we have a good structure of a test molecule. This clearly resolves hydrogen atoms. The R bar becomes low, but it is still high compared with those by X-ray crystallography. We are now studying how to treat scattering factor, dynamical scattering, inelastic scattering, extinction factors, and so on. Okay, finally, let me introduce our effort for automated data collection. So we have developed a system for automated data collection using the deep learning technology. So let me go back to single particle analysis. This is the stage adjustment for single particle data collection. The target protein molecules are embedded in thin amorphous ice inside the carbon holes. However, cross-correlation often fails in locating carbon holes due to thick ice or large contamination. In this example, data were eventually collected from area indicated with white boxes. When the alignment fails, it can be disastrous, as we take 9 to 25 or more image stacks from a hole clustered around a central hole, and or even more stacks for one hole through changing image shift once a stage is moved to a new position. Thus, we develop a deep learning-based real-time object locator using a well-known object detection system, YOLO. Now, hole detection works quite well, and it also works well even for very low contrast images due to thick ice. And you can even detect holes taken at a lower magnification or holes on gold grid. Now, no alignment failure occurs in single particle data collection. Detection of objects is very fast and typically done in 0.06 seconds. Then we extended this approach to Cryo ED data collection. So it also works well to detect crystals. We train with images to distinguish target crystal and ice crystal, contaminated ice crystal. We also trained with diffraction patterns based on the diffraction quality. We can adjust the stage position and evaluate the diffraction patterns just before rotational data acquisition is started. And the program can also detect crystals in low magnification images. The crystal position can be registered for unsupervised rotational data collection by Cryo EM. Now the whole process is almost fully automated. And as I said, we use a well-known object detection system, YOLO. It stands for You Only Look Once. So we named our program You Only EM Once to locate in real time, in short, Yoneo Locker. Anyway, you can download this program suite, including serial EM scripts from our GitHub site if you are interested in. Okay, I skip this. Thank you. The study I introduced today were able to be done through many collaborations and working with many people. I really thank these people. Thank you very much. That's it. Thank you. Sorry.
Thank you, Koji. That was really interesting and gave a real flavor for the diverse range of the research into methodologies you do. You showed us electron diffraction of small molecules.
Yes.
Do you think it will also be important for smaller proteins as well?
Yeah, I think so. Because it is really hard to apply single particle analysis for the small molecules. So it's really important to solve. Yeah.
I mean, we are going to have a discussion between all of us at the end of this. So what I might do for now, if it's okay, is move on to our next.
Please go ahead.
Yeah.
Please go ahead.
Unless, and I will also ask for, so if I could ask the audience that if they wish to ask specific questions about Koji's work, if they can put it, there is an option to ask your question online. And if they can put them there, then we'll come back to them when we start the roundtable discussion. Thank you very much.
Sure.
So our next speaker is Jamie Blazer, who worked in Judy Hurst's lab at the University of Cambridge on mammalian respiratory complex 1, which I'm sure we all remember was one of the first really large complexes solved by Cryo EM right at the outset of the Cryo EM revolution. And I certainly remember all the excitement about that structure. He has been deeply involved in sample preparation methods, including modification. You know, we are Quantifoil, and the modification of the ultrafoil gold grid surfaces is a really exciting part of his work for us. And he's now moved to the University of York, and he's continuing to work on the structure of proteins involved in bioenergetics with Cryo EM, and also on novel methods development to support this. So yeah, I'm really excited to hear from Jamie, and I'll just make you presenter.
Thank you. How do I share? Where's the share button?
Let me just check. You should be able to see.
Ah, there we are.
There you go. You got it. Can you share now? I was hoping Priya would come in for me there. There we go. That's brilliant. We can see your screen. Yeah. Okay.
And so, can everyone see that? Can everyone see my screen? Claire, if you could just say, because the
Yeah, I can
Okay.
see your screen. You're fine.
Wonderful, and I'll just make a note of time. So I was thinking about what to present, and actually my lab have finally got their first preprint, our first independent structure out last week on an electron bifurcating hydrogenase. But I thought, as this was a methods sort of focused meeting, it would be quite interesting to tell you all about the work that I did at this point about 4 or 5 years ago on optimizing complex 1 grids to push the resolution of the complex 1 project. And I think the important thing about this is that the work we did 5 years ago has continued to pay dividends in the resulting 5 years. Obviously, I left to come up here to York and start my own group, so it's other members of Judy's lab who've done that work, but they're using all the methods and all the techniques that I explored when I was a postdoc working with a guy called Vinoth Kumar at the MRC LMB. And so, you know, it just goes to show that when you have a mature Cryo EM project and you've got an important target, spending a lot of time really thinking about how to get the most out of your data, how to get the best grids, how to have the best preparation, can be really, really valuable. And it's a little bit like how Rado published a really nice paper a few months ago detailing similar stuff for GPCRs. So I've called it Taming a Complex Beast, Lessons Learned from Pushing the Resolution of the Mitochondrial Complex 1 Cryo EM project. And just to be clear, this is work I did as a postdoc in Judy Hurst's group. So I'm just going to start by very briefly introducing Complex 1, and then we're going to go into how I optimized the preparation to be in a single state, which was important both to understand our data and to get higher resolution data. How we optimized the grids to improve particle density using pegylated gold. And then finally, very quickly, showing how this work I did 5 years ago has paid dividends over the resulting 5 years with a number of really nice papers. So complex 1 is a member of the mitochondrial electron transport chain. So this is the system that provides our cells with energy by reducing equivalents from central metabolism, passing them to oxygen, pumping protons across the inner membrane, and then those protons come back through ATP synthase to synthesize ATP from ADP. So complex 1 is there on the left. It's the first complex in this chain, and it is a bit of a beast. It's a megadalton. It doesn't have any symmetry. It's got 44 different subunits. It's an integral membrane protein, and it's mammalian, so it isn't very stable. On the other hand, we've got over 60 years of very hard work from various groups to purify this complex in an active state. So it wasn't like I had to optimize anything from scratch. And so when I was getting started, we had what I called at the time the farmyard of mitochondrial complex 1 structures. So 3 groups. There on the left is Judy's group, and they published about a 4 angstrom structure of complex 1 isolated from cow. In the middle, there was a group from IST in Vienna who worked with sheep. And finally, there was a group at Singhar University working with complex 1 isolated from pigs. And the thing that all of these preparations had in common is they're all about 4 angstroms resolution. And we now know from talking to our competitors, friendly competitors, that actually they had a lot of the similar problems as we had, which is unsurprising as these complexes are all essentially identical. But they're all stuck at 4 angstroms, and they were all in a mixture of states, which made functional interpretation rather hard. So the first thing I set about doing was optimizing the preparation to be in a single state. So the 2 main states, we actually had other states which were various forms of the broken enzyme that were breaking on the grid. The main thing we needed to explore was this deactive enzyme structure. And the enzyme has an active and a deactive state, and there were 2 models in those 3 papers I just presented on what the structure of the deactive state actually was. in one model you have a loop becoming unfolded, and that breaks the Q site, which is one of the active sites. And in the other model, actually, that same loop was collapsing across the top of the active site, which also blocked it and stopped it constituting an effective active site. And so the first thing we had to do was start working with a single preparation in a single state so we could explore this in a more rigorous way. And here we used a convenient NADH oxidation assay, which is easy because complex 1 is an NADH oxidation enzyme. But it is worth thinking about if you're at the beginning of a Cryo EM project. If you put broken enzyme on your grids, the only thing that you can image is the broken enzyme. And so you really want to explore the functional properties of your preparation to ensure what you're imaging is really the active, physiologically relevant target. In this case, actually, we could heat isolated mitochondrial membranes to 37 degrees to place them into the deactive state, and they became 95% susceptible to NEM, which is a marker, and we improved the specific activity of the enzyme considerably to match the physiological rate, partly by swapping Simul 7 detergent for DDM detergent. And for those working with membrane proteins, I would say to explore detergents very carefully. There's now a lot of different alternatives to detergents like PeptiDisc and Supposin, and that's very exciting. We tried that quite hard with Complex 1. We were never able to get it into a detergent, anything other than multi-side detergents, possibly because it's got such a elongated shape. And so now we had the complex in a single state, which allowed functional interpretation and also meant we didn't need to do so much classification. Or indeed really any classification other than to remove broken particles in our Cryo EM workflow. And the other thing we did was we moved to a surprising source of proteins. So my colleague Nora Jip was working on mouse models for Complex 1. Originally we were using cow. You can imagine cows as a source of protein are pretty good because they've got very large hearts. Hearts are full of mitochondria. Mice are much smaller. So the prep went from being about 3 milligrams a go to about 0.3 milligrams. So you got 10 times less protein. And also, you know, it was at least 100 times more work to get that protein. But what we found was that comparable preliminary data sets go to about 4.4 angstroms with only 16,000 particles, whereas bovine needed 30-something thousand particles to get to that resolution. And of course, Cryo EM has just got such small sample requirements that even though we only had 0.3 milligrams of protein, that was more than enough to make tons of grids. And I think this really, you know, it really is a new era because mouse models are so important for a lot of diseases and for a lot of targets. You can think about working with native mammalian material from transgenic mice, which could really marry up a lot of cell biology to Cryo EM structural studies. And I think the reason that mouse was such a good source of protein was that the small size of mouse hearts allowed us to get them out of the mouse quite quickly and cool them very quickly because the surface area to volume ratio was much larger, so they cool faster, so they're not sitting around warm as they're and degrading. They get cold fast. And actually, we found that because of that, the mouse enzyme was completely in an active state. So that's what we did to optimize the preparation for the complex to be in a single state, and a single state that refined very well. Now I'm going to talk a little bit more about how we optimize the grids for particle density. So this is not anything I can take credit for. It was a guy called Joel Mayerson who did his PhD between NIH and the MRC Mitochondrial Biology Unit, where I used to be. And we had a problem that our particle density was always just too low. The way that we purified the protein was that we would run a gel filtration column and we would take the protein from the peak of the gel filtration column, and it just seemed like whenever we tried to concentrate it further, it started to aggregate. So we were stuck with about 4 megamil protein. So we needed another method to prepare good cryogrids, and we also wanted to move away from Simul 7, and for some reason that detergent increased the particle density but was bad for activity and actually did increase the number of bad classes that we'd find in classification. So it increased particle density, but not in a way that actually probably provided us with the good particles we needed. So what Joel saw was that pegylation of a gold surface decreases the affinity of the complex for the support so that it is free for imaging in the holes. And this figure is from his paper, from his 2014 Science Reports paper. So here's a comparison. Here are two grids got on our old T12 microscope, and on the left you've got a carbon quantifoil grid, and then on the right you've got a pegylated ultrafoil grid. And you can certainly see that there's many more particles in the pegylated grid, and they're much better dispersed. And I think what's really interesting for me here is we really think about the air-water interface a lot when it comes to the damage and the aggregation that can occur as the grid freezes, or just before that the grid freezes. But here, you know, really, it's showing that the air-water interface in these two situations should be pretty much identical, I would expect. But certainly the grid support itself, whether it's a carbon surface or a pegylated surface, does seem to make a huge difference. And I'll maybe talk through what's happening here with the pegylated surface, because I just realized I skipped that, so sorry about that. The PEG reagent itself, you can see it in the bottom left of the figure below. The way it looks is it's got a thiol group at the end, it's got a 10 carbon alkane linker, and then it's got a PEG6 group, and it's all as one molecule. And thiols or thiolates will form very robust bonds with gold, and the thiol reacts with the gold surface. The alkane linkers pack very tightly, and then all that's exposed into the bulk solution is the PEG group. And obviously a PEG is biocompatible, it's very soluble, it's a lot nicer for proteins there. And the way that we did this is we took gold grids and we incubated them anaerobically in ethanol with the PEG reagent, and that worked very nicely. So here's a real data set. Of course, it's very nice to just show one representative micrograph and compare that to one other representative micrograph, but you can see on the left a quantifoil holy carbon data collection that we did at the very beginning of the project, and then you can see on the right what happened when we used pegylated gold. And so this is at the time a standard collection with maybe 2,000 micrographs, and you can see really many more particles using pegylated gold. What was interesting, and with hindsight, at the time it probably could have been quantified better, but we did also find that the pegylated gold grids seem to explore a slightly larger space of orientations. You've still got slightly preferred orientations. Of course, this isn't anywhere near as bad as some projects which really have bad preferred orientation problems, but they do seem to be a bit more spread out. So there seems to be a greater diversity of orientations when we use pegylated gold compared to carbon quantifoil. And I don't really have an explanation for that, but it can't hurt. So finally, I just want to talk about seeing how this work has paid dividends over the resulting years. So the first structure that Vinoth Kumar solved with Zhao Peng needed about 12 days of cryos time to obtain a saturated data set, and using pegylated gold we got this down to more like 3 days. So that meant we could, using weekend collections, just collect on a single in a single session, and get the resolution and the particle numbers we needed. And then moving to mouse allowed us to move from about 4 angstroms resolution to about 3 angstroms resolution for the active state. And now that with faster detectors, it's possible to resolve below 2.5 angstroms resolution with even shorter collection times, below 2 days, for example. So that's how the project has developed over the years. And I just want to mention the scientific highlights that have resulted from these methods. So this is by and large not my work. I'm on one of these papers, but certainly all of these projects either use the mouse enzyme, or they use pegylated gold grids, or they use both. People in Judy's group have gone on to resolve water molecules, which are key elements in proton pumping mechanisms. They've gone on to understanding quinone binding and reduction, which is an important substrate for complex 1. They've used it to explore disease mutations, again, because the mouse model allows us to use mouse models of transgenic mouse models of disease states, and that's been really important. And they've also used it to resolve candidate drug molecules, anti-cancer agents. And so that's how just taking time to really understand your prep and understand what you can do to get good grids can really, yeah, as I say, pay dividends over the resulting years. So just quick run through of acknowledgements. You can see at the MRC MBU, I was doing this with Judy Hurst and all those co-workers named there, a very tight-knit team. At the MRC LMB, I was working with Kutty Vinoth Kumar, who was really, well, he taught me Cryo EM, so I always owe a big debt to him, and he was really instrumental in this project. And we collected data at EBIC, which is the National Cryo EM facility. So hopefully, yes, I'm just about on time. So I'll stop sharing my screen. Hopefully, is that okay?
That was brilliant. Thank you very much. And again, I'll say, if the audience have questions for Jamie, then if they can write them or raise their hand, that would be fantastic. But we will be having a discussion later when I think we can draw out more of the points. Just on your talk specifically, I noticed, you know, with your discussions about improving those resolutions, you were talking about, you did all three things, so that you were looking at changing the purification, getting a single state, and then adjustments to Cryo EM. With the advent of people looking at multiple conformations on one grid and things like that, do you think that finding a single state will, you know, will remain as important for getting the highest resolutions? Is it, or will things shift towards, will we do that? I guess I'm thinking in a using AI on the grid rather than actually.
Well, what matters is are those states physiologically or mechanistically relevant? So the active-deactive transition I talked about is a physiological mechanism of regulation. But we have other states on the grids that are just the enzyme falling to bits, and we can see that happening. And now I think actually people have, not just in Judy's group, and some of this is published, but certainly have started looking more deeply into that active state and are finding interesting things there. So it really just depends what the states are. And the key is relating the structural data back to the biochemical data so that you can interpret those states to have real findings. So yeah, basically it can be, classification can be either both, is both the great opportunity in Cryo EM, but it can also be a great hindrance if you're classifying particles that aren't relevant.
Okay, thank you. And I think, as I say, we'll just move on to the last of the kind of little introductory talks from Rado Danev, who's a group leader at the University of Tokyo. His lab studies macromolecular proteins by Cryo EM, as do you all, but in particular cell membrane receptors, and he's made significant contributions to methods development in this area, perhaps most recently, as Jamie's already referred to, with a really nice study of the factors influencing high resolution data for GPCR proteins. And it's interesting to see work like that kind of looks to systematically work to improve the ease and efficiency of SPA, because that's what's going to make the difference to make SPA really influential in structure-based drug design, I think, and make it really feasible for structure-based drug design, getting those really systematic workflows worked out for how to do it faster, I guess, is higher throughput. Okay, I'm going to hand over to Rado. If Oh, I can see that he's already there.
Do you see my
Priya
screen?
works in the background. Absolutely. Yeah, Priya works in the background.
Okay, because I
now.
don't see any indication that it's shared. It's dangerous. I shouldn't forget to de-share it afterwards. Anyways, so thanks a lot for the introduction and for the opportunity to chat about the challenges in Cryo EM, especially related to sample preparation. I think this is the area where, as people who have done Cryo EM know, that this is the area where most challenges remain and where we expect most of the advances in the near future, hopefully. So yeah, I'll start with the acknowledgement. So we've worked on GPCRs for 5 years already, together with Patrick Sexton's group at Monash University in Australia. So Patrick and Denise are leading the lab there, and you see some of the current members on the bottom. So Cindy, Matthew, Rachel, and Sarah. And on the top right, you see two of the ex-members of this collaboration, in a sense that they worked in the first couple of years, but now have moved on to other adventures. So Lynn was the member of Patrick's group who did the first Cryo EM optimization of the GPCR samples. So bringing them to taming them down, so to say, and figuring out what works and getting a good homogeneous prep. And then she sent samples to us at the Max Planck Institute of Biochemistry, where Mariam, she was a PhD student in my lab at the time, did the first experiments of Cryo EM, GPCR Cryo EM, and actually managed to get a structure from the first data set she collected. And at the time we were using the faceplate. We tried a few more samples. You see here a list of, I guess, GPCR structures until the beginning of 2019. The red ones were done with the faceplate, and the ones which are also in a box were done in this collaboration with Patrick. So we had quite a good going with these proteins. However, other groups were also pursuing, of course, GPCRs. They are very important pharmacologically and in disease. So they were doing it without the faceplate, and we figured out after I moved to the University of Tokyo, let's try to quantify the contribution of the faceplate. Does it really help? So with the new detectors like the K3 on the Cryos here, the throughput is much higher, and also you get more pixels. So with the K3 we get 40% more image area. And with this higher throughput, we were able to collect data sets from the same sample by changing a single parameter. For example, on the left, you see an image with the faceplate, of course, much higher contrast. The molecules are very easy to see in the background, and the contaminants, of course, are very prominent here. And on the right, about another half of the data set was collected without the faceplate, just with defocus. And by processing those, we compared the final reconstructions, and the faceplate unfortunately produced slightly lower resolution, so 3.0 versus 2.7. And to quantify it even further, we calculated B factors, which is the B factor basically represents the attenuation or the envelope function on the information in Fourier space. So higher B factor means more attenuation of the high resolution components. And here, indeed, the Volta faceplate had a higher B factor than the defocus. So basically, this told us that the faceplate doesn't help for the structural determination of GPCR. So since this data that was collected in the beginning of 2018, but then processed took a few months. So since May 2019, sorry, we haven't used the faceplate anymore. And because this approach worked pretty well by doing this half and half of each data set, so basically it's a real sample, it's a real structural target, but in parallel we can do also methods testing and development. So we also collected half and half with the zero loss energy filtering and no filtering, with and without objective lens aperture, and then we split this data set in additional subsets depending on the defocus magnitude, total exposure, and the grid type. So this is the grand summary of results. The bars which are going below the horizontal axis mean degradation of performance. So the Volta faceplate reduced the resolution by 0.4 ångström in this case, because here we compared subsets of equal size, to be fair. So 100K particles, 0.4 ångström worse with the Volta faceplate. Zero loss energy filtering improved the resolution by 0.25 ångström or so. You see the error bars are quite large though. So, you know, there is quite a bit of variation here probably, but there is a definite effect when the bar is smaller than the error bar is smaller than the bar. Objective lens aperture showed very small positive effect, but with gold grids, it's actually advantageous, because if you have used gold grids, you know that there are reflections, polycrystalline reflections from the support film that sometimes intrude into the image like bright spots. So the aperture stops those typically from entering the image. Defocus is also still a question, but we like to collect with a bit lower defocus than typical, so between 0.5 and 1.5 micron defocus. And total exposure, we noticed a definite positive effect by using more electrons. Basically, I guess getting more low frequency information at at the expense of a bit more longer exposure time. And here is actually the graph that shows the effect of using the gold grid. So H dot represents an individual structure that we determined. So this is from November 2018 until spring of last year, so 1.5 years roughly. We have more than 50 structures in this period, I think. And we were using the standard quantifier grids with the carbon support until August 2019. And then I tried the gold grids for the first time. And it was not because I really believe that you see a difference, but because I ran out of the standard carbon film grid. So it's a bit, a lucky, I guess, misfortune, so to say. So, and on the first experiment, we got this 2.1 angstrom resolution structure of the GLP-1 receptor, which was very surprising because we have done already apoferritin, for example, with standard quantifiers, and we have 1.6 angstrom there. So I didn't expect that, gold grids will make such a huge difference, but they did. So you see, after we adopted the gold grids, everything became definitely or kind of objectively better than before. So on average, I would say 0.5 angstrom improvement in the results. And the orientation of particles, which is represented by the size of these dots. So a smaller dot means a narrower distribution, and this is calculated by Chris Russo's Cryo EM software. The gold grids didn't change much this distribution. So basically, I think it even became a bit narrower because we are using now much thinner eyes, and there, of course, mechanically the particle stays with the long axis parallel to the sample. So, but this didn't affect our ability to get good structures. So we don't have, very strong preferential orientation. There is some bias, but it doesn't interfere with the results. Now, these are the examples from the supplementary materials of this paper. So you see carbon support grid. What is obvious is with the carbon grids, the films are much more fragile, so easier to break during plunging, especially with the larger meshes. And also the distribution of the ice is not as nice as with the gold grids. I think this is probably a combination of factors. It's not the gold film itself that makes things better. It's also the grid material plays a role here. So how the liquid wets the grid and how it is draining during the blotting process. But overall, combining all factors, of course, the gold grids are also a smaller mesh, so they are 300 mesh, so you see the squares are much smaller here. But overall, we see much better and more consistent samples with the gold grids. So in terms of ice distribution, uniformity, and overall, from grid to grid, less variations. If we keep all the parameters the same, they're more consistent. So I think this is probably half of the reason why gold grids improve GPCR so much, if not the majority of the reason, because there is some difference in the motion. Oh, I didn't include this slide here, but anyway, so we also managed to get last year sub-2 angstrom resolution GPCRs. So we have one that is at 1.95 for the receptor and 1.84 for the G protein. We see already clearly the holes in the aromatic rings, and also the pyro ring is showing a tendency of a dimple. I guess if I change the threshold level, it will also pop. The first one was not published. We have a second one, the human CCK1 receptor that was published earlier this year at also 1.95 angstrom resolution. So we are, we are sub-2 angstrom, but not for everything, of course, only for the nicely behaving ternary complexes. More challenging samples, like, for example, receptor-only structures we are trying now, they are still, quite difficult. So I think I have a slide here. Also, without this proprietary nanobody, the resolution is getting a bit worse, so more flexibility, more dynamic. So this is a receptor-only structure, and you see even with gold grids, it's still at 3.2 angstrom resolution. But it's still a great achievement in a sense that we can do inactive state GPCRs in cryo. At least the class B that has a nice extracellular domain, which helps with the assignment of the orientations of the particles. And I'll transition now to a PDF file, which is the supplementary materials of our GPCR performance factors paper, where I'm showing the motion statistics for the different types of grids. So we have here 3 data sets that we analyzed. 2 of them were collected on the standard carbon film quantiphos. You see here the grid maps and some representative squares, and 1 was collected on the gold grid. So if you compare the total displacement, now the motion that Reliant gives is not very useful, because if you have too many frames, it becomes dominated by the noise, frame to frame noise. That's why in the revised version of the paper, we decided to use the displacement. So basically from the start of the movie until the end of the movie, how much motion there is. And you see here that the gold grid, which is the light blue histogram, definitely shows much lower motion than the carbon grid. So definitely they have obvious effect on this. And another thing related to grids, we have another paper which was about testing of the new Cryos we got. It has a K3 camera and a Falcon 4 camera, so we decided to test these kind of back-to-back to see what the result would be. And I also used the time, the chance that I put apofertin in the microscope. It's a rare occurrence nowadays, but I had apofertin, so I tested a few data acquisition strategies, like single image in the center of a hole or 4 images around the periphery of a hole to see what the difference will be. So we have here the so these data sets, they reached 1.3 angstrom resolution, the best one, and the difference between the Falcon and the K3 were negligible. In terms of, I think, DQ, they're pretty well matched. And in terms of motion, the data set 1, which was collected with a single image in the center of each hole, showed the least motion. Unfortunately, in this paper, I still use this motion criteria. So it's not as good as using the displacement. So I did a recalculation of these parameters recently, and I also separated it in X and Y. So this is an X and Y plot. And you see here set 1, which is light blue here, sorry about the colors, is very much focused in the center, this kind of a star cluster here. And the other sets which were collected with the, I'm going back to this, I hope you're not getting too confused, but the other sets which are collected with the 4 images around the periphery of a hole definitely show motion perpendicular to the edge of the hole. So there is still some motion perpendicular to the edge of the hole, which means that indeed there may be a drum-like effect or this kind of a doming effect of the ice. And yeah, I'm currently also real-time collecting a data set. You can see here the microscope is running in the background, and I'm using, of course, ultra aerofoil grids, and we happen to see a nice image coming up. So you see some GPCRs here in the background, and this is actually the grid map of this data set which I collect today. You see it's a really nice, again, very uniform distribution. We have very nice, a very high number of nice looking squares with almost no thicker ice areas. You see these kind of a bit darker, this means thicker ice, and this is one of the maybe practical challenges with the gold grid, is to judge properly the ice thickness. I mean accurately the ice thickness, because the film itself is so dense. Seeing the ice thickness is much more difficult than with the carbon grids. And one kind of I guess comment to quantify is, I noticed in the batch, one of the batch of the grids we have these kind of dirty spots on the grid, and this is not because of our treatment. I've checked the grid itself, so I don't know if this is just maybe a few grids, but yeah, I hope the quality control will improve, and I think Clary said that they're working on this. And to deal with this kind of tricky ice thickness decision, we currently use image J to play with the contrast and also change the palette from grayscale to color scale. So then you see much better variation, the variation in the ice thickness. So this is maybe a suggestion to people if they want to do something like this. And yeah, that's it. So how do I unshare? Maybe Clary can stop my share.
Yeah, I will stop your share. Thank you so much for that. That was fascinating and very cool to see you actually collecting what you're actually collecting right now. Yeah, I will take as ever, if there is a issue with the grids that's of particular concern, then sending us some images is always really helpful, pointing out what you're seeing so that we know we do inspect every grid before it goes out, but obviously when you've got situations like this where you're seeing actually in the electron microscope something happening, very helpful if you can share that with us. I'm going to make myself presenter. We do have a question from Hiroshi Ime for Jamie, but I will wait and we'll come back to that because it is relevant to what we're going to do now. I am sorry, guys. This was organized by Quantifoil and our recognized distributor in Japan, EM Japan. So I am going to talk a little bit about Quantifoil and one of the options that Quantifoil can provide for you for helping with improving sample dispersion on your grids, since we're talking about sample preparation. So first of all, what I've got here is a very simplified little flow diagram about how one might go about collecting data. So when things work, they work. You go through an initial screen, maybe on a Quantifoil, have a look at whether you've got good particle density, move on to a first data collection, and see whether the protein's adopting single well-folded conformations, the conformations that you're looking for, and then look for a high-resolution data collection. And if you meet target quality, you've got your structure, and that's fantastic. But what this morning really is all about, or this afternoon in Japan, I should say, is the red line. When things go wrong, what else can you do? Next. So I'm going to say thank you here to a great paper from Deprina et al. In eLife, which looked at tomography of different of the samples sitting on the grids. So what could be wrong with my particle? And we all know that absorption onto the air-water interface is a major cause of protein aggregation and folding, but also that, as Jamie has in fact already alluded to, you get protein sequestering onto the carbon support and away from the holes, reducing particle density. And the diagram here just shows you how many of the particles are either sitting on the air-water interface in the gold, or are bound to the carbon support in the silver, and therefore effectively not available for use in your SPA experiments. Excuse me, sorry. There we go. So there are many things that we can do to improve dispersion and density, some of which have been mentioned this morning. You can always go back, look at the conditions your protein is kept in. I've set up to most buffer conditions, but of course there's also detergents. You can also look at protein surface modification. We've seen lots of papers recently looking at proteins bound to nanobodies or cross-linking. We could also look at adding protein-friendly surfactants to block access to the air-water interface because it just sits there. Automated sample preparation is becoming an increasing popular choice. An example of that, obviously, Quantifoil sister company SPT LabTech offer the Chameleon, which is one solution. That kind of repeatable, fast plunging can really help with moving protein away from the air-water interface. But I just want to mention this morning an additional ultra-thin layer of continuous carbon can really help with these issues. So proteins absorb onto the additional carbon layer, as Jamie has already mentioned for me very nicely, and that increases. So if you have a continuous thin layer across the top of your holy carbon grid, you can increase particle density in holes, you can improve sample density. one of the useful things about this kind of modification is it's affordable and readily available, and we'll come back to that. It also, many people like it, it offers support for cell growth across the entire grid. If you coat the grid with an ultra-thin layer, it also makes it available for chemical modifications such as biopassivation and pegylation. And it's a very simple way to investigate improving particle density, because you just swap out the grid you're using at the moment for one with UTC. And the UTC, because it is so thin, just 2 nanometers, it really does make a minimal background contribution. So unless you're looking at very small proteins or proteins where contrast is an issue, then there's no issue with your affecting your sample collection. And it has worked now on a wide range of different proteins and sample types. So it was used on complexes, on membrane proteins, on viruses, and on a range of different types of grids. So you can see we've got the R33, the R22, as well as the commonly used R 1.2, 1.3. So, and why use a quantifoil UTC? Because obviously many people add their own UTCs in lab. Well, I would say that we do check every grid before they go out, so that if you use a quantifoil UTC, you know you've got the guarantee of quantifoil's quality and the consistency and reproducibility. We make a lot of these grids, we make them in batches, we make them according to SOPs, so that you can guarantee that you get the same thing time after time. And obviously you're not making them yourself, so you're saving yourself the time of what is essentially a very standard technical job that you can then free up for more interesting work. We're right here for you. If there's a problem with the grids, we can help advise the best way to using them, and we can put them on almost any of our grids. So it's really flexible. So yeah, do consider a UTC. So that is the end of the promotional section. I will stop sharing my screen, and we can move to the roundtable discussion. So I'd like to open up the discussion to everybody now, and I had a few. I'd like to thank all the attendees who wrote in and suggested topics that they would like to see discussed and they'd like to know more about. I've had a look at those and collated them and come up with those that I most want to, that were most commonly requested. And the first one that I'd like to discuss is, I just need to open up the appropriate piece of paper, well, is sample preparation. And really, top tips. So as part of that, I would like to start by saying, Jamie, there was a question to you. Yes, the best practices for sample preparation to improve your dispersion. Firstly, there was a question to you about the pegylated ultrafoil grids, and somebody wanted to know if they were commercially available. So I don't know if you want to address that.
Yeah, the actual pegylation reagent is there's a number of different vendors. We got ours from a company called SensorPath Technologies, but they've closed now. But there are others. I think Sigma's got some of them. And then we just used ultrafoil grids as our grids. So it's just a matter of bubbling some nitrogen through some ethanol to deoxygenate it and putting the grids in, and then ideally putting them in like a sweetie jar or something like that to keep out most of the oxygen. Certainly published protocols that we use, because we have an anaerobic glove box, we just use it. But Joel just used ethanol with nitrogen bubbled through, and as long as there's an excess of the reagent, it's it's not an issue, and the reagent isn't expensive. You can, you know, a few £100 will get you thousands of grids. So the bits are commercially available, but you do need to put them together yourself. But certainly for us, it was worth the effort. It made a big difference.
Okay, thank you. So going on more broadly to best practice in sample preparation, when you have a new protein, do you have a standard way? I mean, and this is opening up to all of you. I mean, especially maybe Rado and Koji, who have, you know, large labs that are seeing, do you have a standard way of treating a new protein? Sorry, you are on mute. I think your micro. Let me have a look at the presenters.
Maybe I can comment while Koji is
Yes.
figuring out the issue. So in our case, always we aim for, depends of course on the protein. So the concentration, for example, nowadays we tend to use higher concentrations than in the past, between 3 and maybe 10 milligrams per milliliter, depending on the size. Smaller proteins seem to require larger, higher concentration to get kind of good distribution of particles in thin ice. And we start with preparing grids using dilutions in half. So 1 grid with the original concentration, then dilute it in half. Second grid, and again in half. So basically 2 times, 4 times, 8 times dilutions. And prepare 4 grids, put those in the microscope, see what it looks like, maybe figure out which concentration works the best. And then if necessary, you know, plunge more grids around this concentration. I typically don't play with the blot time because unless I guess you use something like glycerol or something that changes the density of the buffer. I guess also depends on the detergent, no detergent. There maybe the blotting time plays an important role, but with detergent, longer blotting time seems to work better, at least for GPCRs. Yeah.
Okay.
Certainly what I would possibly add to Rado's answer there, which is very similar to what we do, we are now increasingly working on targets that would be crystallized rather than do cryo EM. So, you know, soluble enzymes that degrade sugars and stuff like that. Because they're so stable, we're actually finding for them, we go really low in concentration, down to have to go to 0.2 to 0.1 megamil protein. And even then you've got almost a carpet completely full. But they're very soluble and stable. Certainly membrane proteins, you need to be, yeah, up where Rado says.
Okay. Koji? Oh dear. No. While he continues to.
I don't know if he's trying to talk to us or somebody behind the camera.
No,
there seems to be an assistant behind the camera.
So while we're talking about that, you mentioned glycerol there, and yes, we saw a paper in the last few days talking about using, which is traditionally been, you know, no go, talking about using glycerol. Yeah, are there any other? So I mean, glycerol obviously would always have been a popular additive to proteins anyway, because it's widely used to stabilize in all sorts of circumstances. I mean, are there any other additives that you think might be interesting?
Well, certainly what I would just quickly say on glycerol, I think the point that Gabe Lander and team were trying to make was that you can work at 10 or 20% glycerol if you need to, but certainly I've had several projects where removing glycerol has helped. And quite often what you find with glycerol is a little bit, maybe like 2 to 5% actually goes a long way in stabilizing the protein and doesn't degrade performance too much. And you theoretically can go up to 20 if your particle's reasonably large. But I think it's not that everyone should just automatically put glycerol in all their buffers, as they often do with gel filtration. You should, just do a few quick experiments and see, because if you can work with less or without it, it is better.
Yeah.
Yeah, what Jamie said. So basically, if you don't need it, so if it's not required by your protein to stabilize it, then I guess in biochemistry people just follow a standard protocol that includes glycerol in the buffer, just as a standard practice, and it doesn't interfere with the other kind of analysis they do. But in Cryo EM, if you can do without it, then definitely don't add it. But if you must have it, then I haven't read yet through the paper, but I guess the gist is that you have to spend a bit more time optimizing the sample, maybe getting as thin as possible ice so that you mitigate the contrast reduction by the glycerol, and yeah, it works in the end.
Yeah, I think one of his points was that the contrast. Okay. So we have a question from Rajiv Singh, who says, I've just gone and sorry, I just shrank the window. He says, what would you recommend if you had a membrane protein complex, which has soluble protein interacting parts? And so you've got membrane protein, you've got complex, and ideally he wants he's got a membrane protein buffer with glycerol and detergent, and soluble protein complexes without glycerol and detergent. That sounds like a very complicated case to me.
Well, certainly in my experience, the membrane protein is going to be the delicate bit. Probably the soluble thing's more robust. So I would start with keeping the membrane protein happy and hope that the soluble is reason, makes do. Particularly if you have a mild detergent, it should be okay. Yeah,
that seems like. So, I mean, so I'm interested in whether, particularly with things like this complex. I mean, obviously, what became popular, if you compare with crystallography in after the mid 20-teens, was using things like stability screens to see if there were things you could add to proteins and protein complexes that would help stabilize. And I'm wondering whether there's a role for stability screening with Cryo EM, even if it were with a narrower range of additives, and whether that would be a useful way forward.
Yeah, definitely. I think what was the name of the method? Holgerstark had this method where they it was like temperature. Was it some kind of, I forgot, but basically it shows the stability. It was a stability test of some sort, and they were using it very successfully with their samples, but I haven't seen it kind of gain popularity with the other groups. There is also another method, which also I forgot the name of it, where you observe the basically the light scattering from evanescent waves. So you deposit your sample on like a very clean, you kind of scrub the surface to be very clean. You put a drop of the sample, and then there is strong illumination from under the glass, and then you see actually the individual molecules absorbed to the glass. I forgot what the name of the machine was, and they were using it in Martin Street at the Max Planck Institute of Biochemistry. They were testing it, and Daniel actually did an online demo on the Discord server, but sorry, I forgot the name. They said that it shows not only the presence of molecules and basically gives you an idea of the concentration, but also you can judge the size of the complex. So I guess if it's a two-component complex, you would see weaker signals from the individual components, and then you would see the assembled one. Jamie, do you know what this was called?
I think that the technique is mass photometry.
Okay.
I
Yeah,
think
something like this, because knowing that the sample is in good condition before even plunging grids would be very helpful, instead of having to plunge grids, then go to the microscope, screen the grids, and then the step of preparing the grids itself adds another possibility for maybe not damaging but really influencing the condition of the sample. So if you know that your sample is in good condition in the solution before plunging, then if after plunging there is an issue, then you figure out how to improve your plunging method, or yeah, something like that. I mean, checkpoints, if we can add more checkpoints without having to go to the final step, cryo, would save a lot of time, I think.
Yeah, I think generally the more you can know about your sample, the better. And the techniques you'll use for that will be very specific. Well, there'll be some general techniques, but also there's probably specific techniques for your particular field. But the more that you can know about your sample before you start doing Cryo EM, the better. So I think the more biochemistry you can do that before you start with Cryo EM, or at least before you start trying to solve structures, the better. Although having said that, increasingly we are using our glaciers to characterize our biochemistry preps. We just do a quick overnight collection and see what's in there, and that can sometimes help to inform the biochemistry. And I suppose that's a slight different in culture, because over the last well, since the resolution revolution, EM time has been so precious and so hard to get hold of. You've really had to know that what you're sending is perfect. Whereas now, at least in the UK, we're getting a lot more microscopes, things like Glacios, a few CryoArm 200s, into most universities, and we can move from being in a position where Cryo EM time is incredibly precious to where it's a little bit more abundant. So I suppose there is a bit of a change there. But I also think there's a change in the field. Historically, people have just needed to get the first structure, and they wanted to get it first because that's how they get their high impact paper. And there's not that many structures left that are completely unknown. And anyway, AlphaFold will probably give you a pretty good answer if it's structured. But that means we're moving into an era where it's going to be much more, what does this mean?
Yeah.
What does this tell us? What can we really understand from this information? And it's going to be much more integrating the structure with other techniques, and it'll become a bit more dynamic.
I think Koji can speak again now, finally.
Yeah. So my account was muted by organizer, not me. Muted by
anyway.
you.
Okay.
So
okay.
What's your question?
Apologies. So we were talking.
Yeah, it's okay. What's your question?
So we were talking originally, and I think it would still be interesting to hear your thoughts on this, about whether when you have a new protein coming into the lab, what your protocol is for handling that.
Yeah. Actually, I asked Hamaguchi-san and Maki-san in my group, and they said it is important to see your sample with a negative staining at first. It's a really good start point, I think. It's a traditional way, but it's a really still good starting point, I think. Also, they said filter paper of your should be stored at the sample chamber of your BitRobot for about 30 minutes to make filter paper wet. It is the Hamaguchi-san said that it is really important for have a good reproducibility of freezing. Yeah, that's a really important point, I think. So how do you think?
Absolutely, yes.
Yeah, I wanted to also mention the negative stain, definitely. So Patrick's group still screen each sample by negative stain, because it shows again at the early stage if they have assembled ternary complexes in this case, and also the distribution of particles gives a very good idea. And yes, on the VitroBot, I put my papers 1 hour before the plunging. So.
Okay, and I am going to ask, so VitroBot has been the traditional answer for plunge freezing. We've lost Koji again, but obviously we're now seeing some developments in automated sample prep instrumentation. I mean, I do have to mention SPT LabTech's Chameleon, but it's not the only solution on the market. Do you feel that will be, I mean, you're talking about things like judging the grids before they go on, the quality of the grids. Obviously, you know, one of the things those automation solutions offer is, you know, some kind of indication before you actually go near a Cryo EM. Do you feel these machines are going to have something to contribute going forward? And you can say no. I'm just.
We have high hopes. It's difficult to express an opinion without seeing or testing one, you know, with seeing how it works with the sample. But definitely, I think the whole community has high hopes about both the Chameleon and the VitroJet and other options that are hopefully coming out soon. So we definitely need other more options for rescuing samples that are stubborn, I think, and reducing the volume of sample that is necessary. Although Cryo EM is already, compared to crystallography, pretty low on the sample volume. Jamie probably with his talk about the amounts they were getting from mice, I guess, with native extracts, especially from mammal sources, I guess it's really important to be able to go down to even not microliters, but how about nanoliters of sample?
Yeah, I would just say for the Chameleon, Steve Munch and Becky Thompson in Leeds have got a very nice paper where they teamed up with Michelle Darrow and colleagues at SPT, and they have got a very nice paper comparing Chameleon, the Leeds' own time-resolved spray device, and the VitroBot, and I think there's another one in there as well. Interestingly, they don't find that much difference in orientations when comparing the VitroJet, the VitroBot, and the spray devices, which is interesting. But they do see more sort of stable, more intact particles. But yeah, I mean, in the long run, in 10 years, I think there's really we should be able to miniaturize everything. You know, when you look at how much protein you lose when you blot, it's something like 99.9%. And then you look at how much of the grid you actually image, and it's something like 1 or 2%, maybe a bit more with the faster detectors. So we're really losing a huge amount of material all the way along. And exactly as Radu said, if you're going to start working with native mammalian material, and I think there's a lot to recommend that approach, it would be really good to just miniaturize the whole thing, but that would also hopefully include miniaturizing the protein purification before. Or maybe we just all do tomography, I don't know.
Okay, maybe we ought. Well, there is a question here from Endeng Perba, which asking about how do you confirm ice thickness, because and he's referring specifically to how important it is for cryotomography.
Tomography or for single particle analysis?
Well, I mean, he doesn't specify. He says, how do you confirm ice thickness on grids, because it's important for people who work with cryotomography. I guess he is working with tomography.
Yeah, actually, you can use energy filtration to measure the ice thickness. Yeah. So that take your image first with energy filtration and without energy filtration, and compare the mean density. Then you can tell the ice thickness precisely from those observations, I think. It's a standard way.
Okay. And I'm going to move it on now, and Koji may have something to say here about this. So we've talked about sample preparation in general, but we had several people asking about cryo EM and smaller proteins. And I mean, obviously at the moment, smaller proteins mostly handled by crystallography. I suspect that going below 50 kilodaltons regularly with Cryo EM will be difficult. I mean, do we see that becoming standard with nanobodies? Would you recommend, I'm referring to Koji because I'm thinking electron diffraction with small proteins.
Yeah, for the single particle reconstruction, you should use FAV fragment or something like that to increase the total mass of your sample. It's a first choice, I think. Also, you can make really small crystals, so that you can see it with electron crystallography. Yeah, that's another choice. I think I recommend. Yeah, of course. Yeah.
Of course.
So how do you think that? So I think the phase plate would work well. How do you think, Rad, at this point?
No, I recently actually am thinking that faceplate wouldn't help with small proteins.
Really?
Yes. Of course, some simulations in the past show that it benefits small proteins, but I think the analysis that were used there were done with, I think they did mostly translational alignments. So basically 2D classification. So for classification steps, it works, it helps definitely to separate particles based on their conformation or say the presence of a small component, a small subunit. But from the point of view of getting high resolution structure of a small particle, first of all, small particle doesn't have much low frequency information. So the very low frequencies are not there because it's small. And second, the orientational alignment. So basically assigning the angles of the particle, they wouldn't be helped by the low frequency information. Therefore, I won't be too optimistic about solving kind of routinely solving sub 50 kilodalton structures with the faceplate, or without faceplate for that matter. As you said, maybe adding additional mass in terms of a fab or some kind of scaffolding protein. There were some papers with scaffolding structures that you attach the small protein to one or multiple sites on this scaffold and then determine the structure of the whole thing. Or now there is this paper last week by was it David Baker's group, that they could design any kind of binder basically to any part of a protein. So my first thought is this would create a renaissance in crystallography again. So if you can design any kind of binder, then basically you can design the thing to crystallize.
So the one thing I have found the faceplate useful for, because we only got our microscope about at the beginning of the year, and we bought the faceplate, so I'm kind of determined that we do something with it, even though Radolf's made his views very clear in an admirable way, in an admirable and very scientific way. But what we do like it for is just screening when you want to see things quickly, and if you want to collect literally a 1 hour data set while you go for lunch, and then because you have got more low spatial frequency information, the particles process very, very fast, align very, very fast, and that can be quite nice. So and actually we're finding with the Falcon 4 the faceplate works really nicely because the Falcon 4 is much faster than the Falcon 3, so you can work in counting mode and still have a nice strong fluence through that point on the faceplate. So that's what we're doing with it.
Yeah. Thank you. And yeah, so much as I hate to say it in a discussion, I'm kind of getting a drift that with sub 50 kilodalton proteins at the moment, crystallography, we have to leave it some space, and maybe that's what we leave it. But unless you move to electron diffraction, in which case Cryo EM wins again. Okay. So I guess you just referred to that, Jamie. So another thing that came up was people asking about how you optimize in an environment where you have limited microscope time and limited computing resources. How do you optimize your experiment?
Is this an environment with a screening 200 kV instrument or
I think
without?
Well, shall we envisage, because these were questions sent in previous to the meeting, what would you suggest with and without? Let's go that way.
Well, certainly, I think it's if you're on a, say, an autoloader equipped 200 kV instrument, I would say that the key is to go broad early on. So explore a big range of concentrations, because that's hard to predict ab initio. Maybe explore a few different sorts of grids, a few additives. If you can work out what additives work nicely beforehand, that's really great. If you're working, I mean, yeah, negative stain on a room temperature microscope can be nice because it's quite easy to do, and is cheap in terms of resources. Although I must admit, for some reason, the respiratory complexes that I work on, I had a number of cases where negative stain didn't work, and then cryogrids did. They just don't like the stains, and I've never really understood why, for those particular class of enzymes. But I think it is really useful for other projects if you don't have access to a high-end instrument, certainly. Yeah. I guess the more you can know about the biochemistry before you start the Cryo EM, the more tools you'll have to try things, particularly with additives like magnesium sulfate or different detergents and stuff like that.
Yeah. I guess, and do you see a way through, thinking about widening access to Cryo EM? Could you see a way through, and I'd be interested in Koji's view on this actually, where one might have in a home lab some sort of plunge freezing and storage facility, and then you might use a central facility like Spring 8 offers, and maybe the new Amazon Web Services by the hour computing support. Could you see more ordinary molecular biology labs managing to do significant structure in that kind of environment?
I don't think so. Because the transfer of your huge data takes a really long time. So the people don't usually use such a cloud service yet. So I also asked this question to Hamaguchi-san in my lab. So to improve your analysis, image analysis. So the selection image should be carefully done after determination of CTF parameters. So that you should see the gallery of CTF link patterns, and then select good ones manually. It's a really good point, I think. I heard so. Yeah, actually, I also do this for selection of images. That improve your usable data, I think. So you can extract much information from such limited data, I think, we think. So that.
What I would just add to that as well is I think we've been accessing the UK national facility through lockdown completely remotely, and probably before, if you told me that, I'd be like, well, you know, Cryo EM time's so precious, you may as well just get yourself down there, because it, you know, just in case anything happens. But coronavirus has forced us to access it remotely, and I will not be going back to driving down to the national facility, even though it's a 3-hour drive. And we just saw that, you know, Rado showed us his remote collection. I think these days your grids will need to travel, but there's really no need for you to, for your high-end collections. Koji is completely correct that waiting for the data to transfer back only really works if you're doing for your high-end collections. If every bit of screening you need to move the data, that's going to get really tortuous.
Yeah, Yeah, no, good points. But you see things going remote. Do you? I know that the crystallography national facilities actively preferred it when people stopped going in and started collecting remote 5-10 years ago, because they said the users just broke things. Is that also true for you guys?
Yeah, yeah. So our place, Spring 8, is a really remote place. It's a really far from the city. So the remote data collection is really, really common, already common in crystallography. And also we can do Cryo EM data collection remotely. It's really well, yeah, yeah.
And
yeah, remote collection is very, very helpful, and I think will save a lot of resources in terms of time, travel and so on. Although I would say some people, for example me, prefer to sit at the microscope from time to time and to or maybe to go to a beamline and see how things are going. Just sitting at your desk and expecting the data is not much fun. And the same as with the online meetings, they are also okay, but not as great as in-person meeting, right? So it's the same with the machine. Sometimes in person is better or more fun.
That's true. I completely agree with Ralph says. Yeah.
Okay.
Yeah.
Okay.
And I think for screening, we really need, I don't know how it is in Japan, but really need to make the case to funding bodies that universities and research institutes need their own screening instruments, 200 kV autoloader, or I don't know what the autoloader for the cryo arm is called, but that equivalent. So we really need to keep making the case for that. And I think an important point of making the case for screening instruments to be in every university is the fact that the lower sample requirements and the fact that you aren't limited with crystallization does mean that we can imagine being many more users beyond just the structural biology community in, for instance, the molecular biology and cell biology community, where people have much less protein, but the protein probably comes from a more interesting non-recombinant source. And so a big thing we're trying to do at York is get sort of non-traditional users involved. And obviously they need a lot more help because they have a different expertise. And I think for initial project optimization, you have to have it so that you can be on the microscope rapidly and you can very quickly move between the biochemistry and using the microscope.
Yeah. And another question we have is whether you have, and again, this kind of specialist suggestions for complexes as opposed to anything else. And I guess, those are questions around making sure that you still have the entire the complex complete and things like that. Are there any special things you do when it's a complex rather than a single protein?
Yeah, we usually must do the size exclusion chromatography analysis. Yeah, so it is really important to see the sample is homogeneous state, so that it's really important to do the size exclusion chromatography, even for the complex molecules, and also the tri-gradient fixation called graphics for the such a complex molecules. Yeah, I think it's a standard way. I think.
Yeah, and Koji said exactly what I would say in terms of getting size exclusion working nicely. And what I would just add to that as well is we use those 2.6 milliliter gel filtration columns a lot. And you can do a lot of optimization, and they are small, but peak fractions are maybe 20 microliters, and 20 microliters is enough to make 8 or 10 grids. So, you can miniaturize things and quite rapidly explore conditions to make sure your complex is eluting nicely from the gel filtration column. And those, we find those little columns really powerful for that.
And be very gentle with your sample. So depending on the type of complex, they can be, they don't like to be disturbed too much. So
yeah,
we found, I was not a biochemist, but I remember in this GPCR project once, in the beginning, I used to, I don't know why, when I pick up sample from the tube, from the aliquot, I did it kind of several times, you know, kind of quickly mixing it, thinking that this will improve it. But I think this actually caused, in a couple of cases, the things to disassemble, the shear forces in the liquid. So, but you know, people who do biochemistry know these things very well.
yes. I think those of us who come from more chemistry and physics backgrounds have to learn how gentle you have to be with biological samples.
And what Koji suggested, yeah, fixation is, I think, a way to keep things together. If something is really intermittent and you know, the complex can happen only in particular conditions. Nowadays, we plunge in very high humidity, so in principle, evaporation shouldn't be a factor. But I think even in these conditions, when the film becomes ultra thin, the surface to volume ratio is so high that even a very small evaporation rate can still cause changes in the buffer composition, just a kind of milliseconds maybe before the thing is plunged. And some things that are very sensitive. I remember Rasmus Schroeder was visiting us 20 years ago, and he was plunging their samples only in their place because the actin myosin, they were trying to observe actin decorated with myosin. This system was very, very easy to fall apart if I think the salt concentration or the pH changed by a little bit. So fixation would probably help in these cases.
Yeah. My only concern with fixation, which draws us on to another topic that came up a lot in people's questions, was how does that affect conformational flexibility, or do you ever have concerns that it may be affecting the native conformation of the complexes?
I wouldn't say that it would affect. What it could do is stabilize, say, a dynamic system like we observe nowadays with the GPCRs. Always we do this 3D variability analysis in CryoSpark, which always show that the G protein and the receptor have kind of a motion because their binding is not that strong, and there is always some kind of motion there. So I guess if we fix those, this will kind of arrest such flexibilities in a complex. So this may cause you know, people having less information, in a sense that they will get one maybe rigid structure in the end, but they won't be able to analyze further for such you know, spectrum of motion or something like this. Although it can be a challenge on the other hand. So it depends on the case, I guess.
Yeah. I actually see. Go on.
I'm curious nowadays with the resolutions we are achieving. Has somebody seen the cross links in their reconstruction? Say if you go sub 2 angstrom, I guess at some point we should be able to start seeing the cross links and maybe judging how much they affect the structure.
Mm-hmm. Koji, have you ever? I mean, you've showed some very high resolution structures. Have you ever seen a cross link?
No, we've not tried yet. So that yeah, it's a good point, I think, so that we can check both such a we can probably analyze such a very details with a higher resolution single particle reconstruction. It's a good. Well, it's a good point, I think. Yeah, we can do probably in the near future. Yeah.
Yeah, that would be very exciting. I think one of the most exciting things going on in Cryo EM at the moment actually is its ability to see different conformations. Do you guys see? So I think that the difficulty with it at the moment is understanding in what order the different conformations appear, because obviously you lose your time dimension how do you guys see that developing? Will that be AI led, or yeah, I'm just interested. How do you see that field of looking at and how far can we push looking at conformational flexibilities?
Yeah, there are lots of really nice programs. One of them are using AI called 3D Flex, developed by the group for the CryoSpark. It's the same group as the CryoSpark developing team. I think it's a really good program. I think so. Another good software is CryoDragon. It also uses a kind of AI. So that yeah, I think you can choose these sophisticated program to analyze your motion. And yeah, well.
Well, as you mentioned, oh, sorry, Jamie, go ahead.
No, Rado, you go. I think one of the things is that as we get more sensitive detectors, we will see the natural motions of complexes and proteins that we know from NMR will occur. And it will be important to distinguish between the breathing motions that proteins go through and mechanistically relevant conformational changes. And I'm pretty certain that we'll soon see some papers that are sort of detecting motions that they overinterpret as without any mechanistic information or molecular dynamic simulation to relate some of the conformations that they'll see to enzyme mechanism. Rado, you go now. Yeah,
so basically what we see with GPCRs, at least in the beginning, I thought it's just the Brownian, fluctuations, thermal fluctuations of the complexes, any molecule. And Jamie said every molecule has thermal motions, and we see those. But physiologically or biochemically, which one of them, which modes of them are important? We had in a recent paper about two structures, semaglutide and tascoglutide with the GLP-1 receptor. Both of them showed almost identical static structures. So the consensus map, showed no differences in the bindings or the action of the two ligands, but the dynamic analysis showed that semaglutide had a larger motion. So the receptor kind of some of the helices were folding out, and this also could explain, at least the hypothesis is this may explain why semaglutide is showing different signaling bias, or why, although the static structure seems to be identical, and it's also looks like almost the same as the native ligand, why is it acting in a different way? So dynamics play a role, especially with receptors, because everything there is dynamic, I guess, and very intermittent. All these things that we see now as static, they happen, I guess, in nanoseconds probably, or microseconds. I don't know what the time scales are there, but and as Jamie said, untangling the thermal motions from motions which are actually related to the function of the molecule will be really important. And yeah, we have to be careful not to overinterpret or be overly enthusiastic in the claims of what we see is maybe related to the function. It's beautiful though, for example, the ATP synthase, or these rotor type molecules, they are obviously showing very nice motions which are related to their function, but this is rare. This is not as beautiful in other complexes, I guess. So.
Yeah, I think motion as clear as that is rare, yes. And I do think Jamie has a very valid point about, when is it Brownian motion, when is it really mechanistically important? But I guess it's fantastic to have the opportunity to start discovering these things. It's probably the first time we've seen genuinely recorded motion. What we have to figure out how to put it together, motions, but different states like this at this level. So I think
actually,
it's really
your comment, your comment reminded me that it's all Brownian motion. I mean, most of it, And what happens is when there is some particular function, it's a biased Brownian motion. So it would be important maybe to distinguish the change of the character of the Brownian motion from, freely, something flopping around unrestricted. And then when there is a biased Brownian motion, if we can distinguish that, then it will be a result, a real result in a sense.
Yeah, very exciting. And I think, I guess we're going to see some, so just to wrap it up, because I'm looking at the time and we're, I guess we're going to see we've talked almost exclusively about SPA, we're going to see move to tomography, but I think that this aspect of being able to see motion is in some ways where, as Jamie referred to earlier, that we've seen the proteins. What are we going to do? We're going to be looking at mechanism, and I think this kind of analysis of the conformational states of the protein is where SPA will go. I don't know if you guys agree.
Yeah, and in relation to dynamics, also the time resolved sample prep, which I didn't mention, but currently what the current lighting systems can do is millisecond level, unless you go to some kind of uncaging of something with light, I guess. It would be nice if we can shorten the time or allow us to have microseconds or even nanoseconds. That would be great. Of course, we couldn't dream of pico and femtoseconds like the free electron laser people, but why not? I wonder though, if you have, for example, a photo system and after you freeze it, does it still react to light radiation? Can you induce some small conformational changes even in the frozen state? Maybe Onekuro-san can comment on this. If you have light photo system and you irradiate it after freezing it, after vitrification, will it show structural changes or not?
Yeah. So I want to comment on the XFL study. So the XFL can be used for the really short motions. Its time scale is different from the Cryo EM motion. It's in the Cryo EM, so that you can see huge motion. It's a relatively long time motion, I think. So that it's a two methods are completely different in this point, I think. So that you can use both methods to study much detail on these dynamics of the working molecules. Yeah, it's my comment. So sorry, I can't understand what Rad said.
I was asking about the photo system. You had this high structure of photo system to 1.95 angstrom structure.
Yeah.
Photo system too. So the photo system, it's light sensitive, right? And they do this free electron laser studies where they do pump and probe experiments. So when you freeze it after vitrification, is it still light sensitive? Are there some changes in it if you irradiate it with light after freezing, or no more changes?
Yeah, actually, the people think so, but the another issues came up. It's a radiation damage. It's a reaction center is composed of manganese clusters or something like that. It's really weak for the radiation damage. It's really difficult to see such clusters even with Cryo EM, because Cryo EM use a huge electron dose. Even if you can use a dose weighting scheme, but it's still radiation damage effect is still there, I think. Yeah, it's really difficult to analyze. So that you can probably use first one or two movie frames to analyze these. You have to use these frames. Yeah.
Okay. Right. So it is 9 o'clock on the dot. So 9 a.m. here, 5 p.m. in Japan on the dot.
Yeah.
So I think perhaps it's been a really interesting talk. I've really enjoyed actually the roundtable section of this and chatting over where we're going. Thank you all so much. Thank you for everybody listening in who's been attending. Thank you guys, especially for coming and sharing with us today. It's been brilliant. Thank you so much.
Thank you.
Thank you.
Thank you.
Bye.
Bye bye.
Bye.
Bye.